Maybe the first question, life sciences in your field. How you see the new developments, or how important are the new developments for the research you are doing?
I think it's a great time to actually be able to do research now. I'm interested in intercellular symbiosis, and intracellular has cellular to it, and now we can finally work at the cellular scale in an organism perspective. So we can take a placozoan and we have the single cell perspective, and that for me it would not be possible 20 years ago. So I think the life sciences has taken off. The kind of research I'm doing, but also in the medical field, in translational approaches, things are going on now because of the cellular perspective that we can take. That is amazing.
I mean, you are part of developing new technologies in the life sciences. Can we go a bit more specific in what happens in the cell, and technologies which allow to see what is in the cell?
So we have two things that we are interested in, and they go together. One thing is expression of genes and the proteins that are connected to that. And the other thing is, now we know what is expressed. Well, where is it actually acting? So we can now combine these two things. On one hand, where things are expressed, which cells, which cell types, which symbionts express things. There we can actually combine host expression and symbiont expression. So we have that dual layer. And then we have the massive advances in imaging technology, where we can then actually visualize where these things are happening inside of the cell.
I mean, are we at the end of the development, or what are challenges with these new technologies?
We are just opened the door to molecular resolution at the cellular level. The massive data is the problem. I mean, that's the challenge. I mean, this has to be stored, this has to be processed, this has to be perfectly analyzed, and the tools are just developing. I mean, to register the data in 3D, to find consecutive structures from one layer to the next as you move through a cell, for example. This is something that is just emerging, that we can do that in a fully automated scale.
What is for you special on and with being member of Key Life Sciences?
I think Key Life Science is a unique opportunity of people getting together that are very much aligned to what I would like to do and how I think about the interaction between organisms. And this is unique, I think, in all over Germany and much over Europe, that you have people that come from different fields, but that are always interested that there are multiple partners, and how do these partners interact, how they actually then at an organism scale or at the population scale even manage to adapt, manage to survive, and also to, let's say, create something new. I mean, these emerging properties that these major organisms have, this is something that Key Life Science is so much focused about. For me, this is a dream come true to be part of it.
Or are there specific aspects, particularly with your own research, which you would like to have implemented or added to Key Life Sciences?
So we definitely have a core competency in imaging, both at the technical level and also in the people that we have. But for several approaches, we need to collaborate. The lucky thing is that we can collaborate in close vicinity. So Kiel Life Sciences in a regional context, and say, okay, yeah, we do our certain focused things in Kiel, but we have our specific partners that we better integrate, that can be platform providers, that can be knowledge providers, that can be training units.
That's an excellent idea, Harald. Maybe we elaborate a little bit on that. And it's a great idea to associate with KLS, but I also think, could we do something actively with Bremen and the Max Planck?
Some of the DESY capabilities are imaging capabilities, like the FIB-SEM, where you can in 3D go through very detailed intracellular structures. that do not exist in Kiel, and I also don't. It doesn't make sense to in parallel develop them in Kiel, because there is another level of investment compared to the metabolomics that is already very high. So we're talking about several millions for each of these machines, and at the same time you also need the user space and you need the professionals behind that. That's where I see the imaging capabilities of the DAISY to provide the analysis from the single cell to the organelle to the molecular level. This is where the DAISY comes in, I think.
Harald, would you share with us some of your plans which you are for your research in the next 5 years?
I'm a big Star Wars fan, and the funny thing is that in Star Wars, the Force is actually coming by from midichlorians. And now in my work, I work with bacteria that are called midichloriaceae. So some other Star Wars fan actually named them after these Star Wars ideas. And I was wondering, in my midichloriaceae, what is their force? So what are they actually doing? And the interesting thing is, we described this midichloriaceae. They are intracellular symbionts that can sit inside organelles like the mitochondrium or the endoplasmic reticulum. And these intracellular symbionts we found in a lot of different animals. So we found them in placozoans first, but then also in your Hydra. We also have them in single-celled algae. And so what makes them so successful? Why can they be in so many animals? And looking at this, I found that they are actually nutritional helpers. So they're kind of like blacksmiths handing out enzymes. And that's what I'm looking into right now. What role do they play in all of their partners? So we've been starting now to look at what kind of enzymes they have. And that is what I would like to explore. This role, this force, what is the role in different ecological settings? So it's if it's in a like your Hydra, for example, that are predators that definitely don't eat laminin that much. In corals, where corals, they have an autotrophic symbiosis, but they also filter feed a lot. And this filter feeding, that's exactly generating a lot of laminin input for corals. In algae, I mean, the single-celled algae, we have now in the lab, they also have these symbionts, and these symbionts have these enzymes. So looking at the force of the midichloriaceae from single-celled animals to very simple placozoan model, Hydra model to corals. This is the main research area I'm now working at.
How you see the life sciences developing, Indy? We talked about it already at the beginning, but where will be the life sciences in general, and maybe the life sciences in Kiel, or where should they be in 5 years or a bit longer? Considering what we have said before.
So different parts of the life sciences are necessary to actually conduct the studies and the level of studies that we are doing. The calcium imaging, for example, that you are using. This is a technology that comes in from another part of life sciences, but we're using it then also in a symbiosis context. And these are integrating different pieces. But this also needs to be integrated already at the level of education. So we cannot just teach a biologist the old school tools, and here is the qPCR, and you do everything with qPCR. That will not work. We have to educate them so that they can actually at least have an understanding what tools are available, where are the best people to collaborate with. But we also really have to learn that modern life sciences is integrative life sciences.